Hello and welcome to everyone to this live stream today. We will be talking about antibody libraries for animal health research. And antibody libraries, they have become a quite interesting and popular topic because they're used for a lot of different applications, no matter where you are, no matter in which kind of application field you will probably have to deal with antibodies at a certain point. So without further ado, let's go into the presentation and start talking about some contents. So let me switch this on. And there I am. So wonderful. Before we talk about antibody libraries, a couple of words about us. At Lab4Pose, we are on a mission to help develop new therapeutics and new diagnostic tools, especially for companion animals, but not only. And on top of this, we want to help reducing animal testing in human health research. And we do our little piece by supporting researchers with dissociated tumor cells, biospecimens, especially from animals. So here we have dissociated tumor cells from cats and dogs. We have healthy cells from a wider variety of animals. We have frozen tissues and formalin fixed, paraffin embedded material. We have blood and biofluids. And then from blood, obviously, all the different isolated cells. And on top of this, we have a portfolio of some complementary products, for example, antibodies, ELISAs, growth factors, and cytokines for more than 16 different species. Finally, we collaborate with Altis Biosystems, and we help them bring in the human gut models to developers, to pharmaceutical companies. And human gut models are obviously an important building block of organ-on-chip systems, alternative methods that would help also reducing the use of animals in human drug development. Okay, so the first question is, what are antibody libraries? What are antibody libraries for? Antibody libraries, as the name says, is just a collection of antibodies that are that's been designed for a particular scope, for a particular goal. And obviously, it depends a little bit from the type of library, from the kind of application. What is the quality of the library in terms of formulation, in terms of design of the library itself, or the composition, and depending a little bit exactly from the aim, we will have different kind of designs. But in general, an antibody library is designed to maximize diversity so that the researcher would have already a quite wide range of antibodies without having to produce that by themselves. They just have to use them for the screenings. But there will be obviously some differences if I'm designing an antibody library for diagnostics or for therapeutics, or if I want to use this just for R&D tools. Maybe I want to produce kits or antibodies that will be used for a Western blot, for a FACS, a flow cytometry, for an ELISA, for an immunoprecipitation, or maybe I want to do just an inhibition assay. So there will be a lot of differences there. Not just in what are the kind of antibodies that I can find in this library, but also in what is the formulation of the antibody itself. Obviously, in my vial, I don't just have the antibody, but I would also have preservation molecules or other compounds that I would use to keep the antibody in the right shape, in the right folding, and so on and so forth. And then obviously On top of this, I will need to understand which one are my positive controls, which one are my negative controls. So these are also important applications for an antibody library. And the funny fact is that antibody libraries have been changing a lot in the past because the amount of targets has been changing a lot. The kind of approaches that are nowadays available, they are much broader than before. So one of the classical ways of developing targets, especially in therapeutic fields or in the diagnostic field, was to say, okay, I will take a marker that is on the surface and I will design an antibody against that. And obviously here we can discuss if a monoclonal, polyclonal antibody, what else would be, better suited. But that was the original approach. There's a marker and I create an antibody for that. Now, we evolved a little bit in the technique, and so there's a lot more things that we're able to do with these antibodies. We're much better able to define their characteristics, to study, for example, their binding site, their efficacy, binding efficiency, and a lot of other parameters. So we can also use them to do real functions. And one of the things, for example, is to block the dimerization of receptors, and basically avoiding that there's a signal cascade. But we can also design other kind of antibodies that have a particular function, for example, a blocking, an inhibition, neutralization function, as it is used, for example, for CAR T cells, or as a complementary kind of treatment regimen for large molecules, for CAR T cells, and so on. We would say, okay, we want to block the mechanism, the resistance, immunoresistance mechanism that the tumor cells would develop. So they normally produce this PD-L1 immune checkpoint proteins to interfere with the activation of the immune cells, and so we are going to block the blocker. And these are another kind of target that is becoming a lot used now in the clinic. Then we have all the sorts of intracellular tumor-associated antigens, and here we have oncofetal proteins, we have novel targets, really specific for a particular set, tumor type. Maybe sometimes there are also targets that would be expressed normally in other tissues, but are expressed here on this tumor type, and so they're recognized as antigens. And this is also now obviously a quite new evolution, because there's like intracellular delivery systems that are becoming more popular. Obviously, these kind of applications for, let's say, diagnostic based on paraffin slides, and so it was already used since a long time. But as a therapeutic instrument, not really. And this we are growing a lot. And then another point that is becoming even more interesting is, okay, these these intracellular markers, they are being processed then by the proteasome machinery that is normally working to basically keep our cells safe and make sure that the proteins are properly folded, they are working, and so just to be sure, they just remove proteins after a while, and they process them, and they make sure that they are cut into pieces. And this is also part of the immune mechanism of our cells, because these pieces of proteins, the different epitopes, are then loaded on these MHC complexes that every cell has. So the MHC type 1 in this case, and these are exposed then to the immune system. So the immune system can say, okay, there's something wrong in this cell or not. Obviously, the if there's a tumor, in most of the cases, this is really difficult for the immune system to detect. But in case of a viral antigen, this is the reason why nature developed this MHC. So we can also use this kind of mechanism to detect oncogenes or oncoproteins inside the cell. So this is also one of the new approaches where what I'm targeting is not the protein directly, but the complex between the piece of the protein, the specific epitope of that particular antigen loaded on the MHC. And this is opening a whole new world of possibilities. Now, having said that, there's different kind of techniques to develop these libraries, and we will briefly go through it. There are obviously some techniques that are a little bit more classical techniques, and other techniques that are relatively new. Or, I mean, here we treat just 4 of them. There are other techniques that are much more, let's say, modern, synthetic method, and so on and so forth, but they're also not that widespread. And so here we have to make a good balance between feasibility and scalability. So on the one side, the first technique is hybridoma. In a hybridoma, I would basically immunize a mouse by injecting a particular target, or, for example, a cell lysate. If I want, for example, to develop a library for a particular tumor type in dog, for example, then I would take the tumor, will lyse it, and I know that in there I would have also my target, and this is what I inject. The mouse will develop B cells, well, not develop, they're still there, but the B cells that would recognize this kind of target, they would proliferate and start producing antibodies. And so we can then fish the B cells out, fuse them with the myeloma cell line, and basically, after a selection with a HAT medium, I would have kind of B cells that continuously produce antibodies. And so after this, we make a single cell dilution. Would have my single B cell clones or hybridoma clones at this point in a well, and then I can make my screening, cloning, characterizing what are the kind of antibodies. In this kind of application, obviously, I'm starting from a mouse antibody. So if I want to use this for therapeutic applications, for example, if I want to develop antibodies for dogs, for cats, and so on, I will have to change the sequences that are mouse specific and adapt them to the dog or cat. Another technique is phage display. And phage display is also gaining a lot of traction at the moment. Most of the time we start with PBMCs from, for example, animals that would have a particular disease or a particular tumor, and we would take the PBMC, isolate the RNA, and from the RNA we would produce the cDNA. Sorry. From the cDNA, we would clone the immunoglobulin genes into a display vector, and with this display vector, we would transform E. coli. Now, the infected E. coli would be able to present different different parts of the immunoglobulins, but the reality is that what we want to do is package these into a phage, an helper phage, that would present these fragments of antibodies on the surface. So we would infect the E. coli with a helper phage. They would lyse the cells, incorporate the cloned DNA, produce, or basically the fragments, and incorporate them on the capside. Now, the next step is I would take an immune tube or a plate, and I will coat this with the target, if I know the target, or with a cell lysate. For example, I would take, as the example before, I would take tumor cells from, I don't know, colon carcinoma, dog colon carcinoma, and I would use that, would dice that and use that to get my my lysate. I would coat with the plate and then incubate the phages on top of this. Then I can wash this, and I know that the more specific ones will remain attached, and then I can characterize them and continue the cycle. The third method that is most commonly used is yeast display. In yeast display, this is relatively similar to the previous method. Now, in this case, I will clone the immunoglobulin gene into a display vector and transform with that a yeast, for example, Cerevisia, and I will then induce the display on the yeast cell, on the cells itself, and then I will incubate this with a tagged target. So my target with, for example, a fluorescent tag, and then I can sort them with FACS. So I would have then my yeast colonies on one, or fluorescent yeast on one side, and the non-tag on the other. So this way I can sort which one are specific and which one not. And then I can characterize them, introduce some variation to have more specific clones, and basically recontinue the cycle. The last method is the ribosome display. And this is also a quite interesting technique. In this case, I start with the immunoglobulin gene, cloning into a DNA library. And then I will have to make sure that in my library, in my whatever I'm using, I have a promoter, the ribosome binding site, the start codon, the gene of interest. And then between the gene of interest and the stop codon, I would have to insert a spacer sequence. And this is to prevent ribosome dissociation. This is really important for the technique, for the step afterwards, because we then take this DNA and transcribe this first in vitro. So with a transcriptase, then I will have my messenger RNA, and then I can make basically a cell-free in vitro translation system, incubating the mRNA, the ribosomes, and obviously the amino acids. And this way I have basically that the mRNA, because of this spacer sequence, cannot detach from the ribosome. And the same for the antibody. So it doesn't come to the stop codon, so the antibody is still attached to the ribosome. And so I basically have that antibody RNA are bound together by this ribosome, and so I can identify which RNA is connected is producing which kind of antibody, which is obviously great because it spares a lot of work. Then I would stabilize this complex, immobilize my target on magnetic beads, for example, or microtubule plate, incubate the complexes and wash away the unspecific binding ones. Then I can just isolate the mRNA, produce cDNA, characterize, and recontinue the cycle. And that was all for today, talking about antibody libraries for animal health research and all the other possible applications. If you are looking for primary samples, especially from companion animals, PBMCs, or if you're looking for primary tumor cells to produce your libraries, then you can contact us at the contact here, the email that you find here, info@lab4pose.com, or at the telephone number that you can find here. We'll be happy to support you as quickly as possible. So having said that, thank you very much for being on this live stream, and see you in the next one.